and welcome to today's session of e-learning. It's for third year students of Botany, Semester 6, BOC 109, course title Molecular Biology and Genetic Engineering, which belongs to the Unit 1, Nucleic Acid, Carriers of Genetic Information. The module name, DNA, RNA as a Genetic Material. I, Dr. Maria A. D'Souza, Assistant Professor, St. Xavier's College, MAPSA, Goa, will present to you this module. The outline of the module, the experiments, observations, conclusions that led to the discovery of DNA, RNA as a genetic material. We will see Frederick Griffith, Hershey and Chase, Avery McLeod and McCarthy, Frankel Conrad and Singer's experiments. Once the student completes this module, he'll be able to explain the experiments that led to the discovery of DNA, RNA as the genetic material and illustrate the steps involved in these experiments. To begin with, we will first see Frederick Griffith's experiment in the year 1928, which is the discovery of the transforming principle. In this, Frederick Griffith took 2 strains of Streptococcus pneumoniae, which infects mice and causes pneumonia and ultimately the death. He took 2 strains, which is S3, which is the smooth, and R2, which is the rough strain. You can see on the pictures, S strain is the virulent form and it has the presence of a capsular gelatinous polysaccharide coating which contains O antigen through which it recognizes the cell it infects. Whereas R2, that is the rough strain, is a mutant variety. It's non-virulent and it lacks the capsular gelatinous polysaccharide coating. In his experimental setup, he set up 4 experiments, wherein he used in the first one, rough strain, the non-virulent form, and he injected into the mice and the mouse lived. Whereas the smooth strain, which is a virulent strain, which is causing the death of the mice once it is injected. In the third set, he took the same smooth strain, but he heat killed the smooth strain and he injected into the mouse and the mouse lived. Whereas in the last set, he used the rough strain and the heat killed smooth strain. So, students, it must have been seen that the mouse lives because individually in the rough strain and the heat killed strain, the mouse lived. But he saw an unusual observation. He saw that the mouse dies. So what Griffith did, he did the post-mortem analysis of the blood of these dead mice, the 4th set, and he also recovered the live S strain bacteria from it and grew this bacterium on a culture plate, and when grown, they produced shiny colonies and some produced rough colonies, showing that the transformed one give rise to the virulent ones, showing that the transformation was permanent. He concluded that the heat-killed virulent S3 type strain must have caused a transformation and transferred some factor or component of the dead virulent form to the live non-virulent type R2 strain, which empowered them with the ability to produce the polysaccharide capsule and make them virulent type. He thus hypothesized that it is a transforming factor and the phenomenon as transforming principle. Although his results were significant, his observations could not identify the biochemical nature of this transforming principle. Following Frederick Griffith, it was Alfred Hershey and Martha Chase who did the Blanders experiment. In the year 1952, using bacteriophage T2, and they did the isotopic studies. Used bacteriophages which contains genes that allow it to replicate in E. coli bacterium. They attach deliver its genetic material into the bacterial cell, where it generates more virus particles. Since the phage, that is the bacteriophage, is composed of DNA and proteins only, its genes must be made up of one of these substances. To function as genes, one of these substances has to enter into E. coli. By utilizing this thought, the experiment was performed. And they did the first step. They grew the bacteriophages on an E. coli medium containing radioactive isotopes, that is P32 and S35. So the first step was labeling. And the viruses were labeled. Labeled DNA had phosphorus 332, whereas the phage capsid has S35. Then these radioactively labeled phages were added to an unlabeled E. coli culture. They were allowed to infect the bacterial cell for a sufficient amount of time. Once that was done, they did the blending to remove the protein coats of infecting viruses from the bacterial surfaces. Following, they performed centrifugation, and the centrifuge fractions were analyzed for distribution of radioactive substances P32 and S35. They observed that the bacteria infected with viruses that have radioactive protein were not radioactive, whereas the bacteria infected with the virus that have radioactive DNA were radioactive. This is the flow chart to show you labeling, infection, blending, centrifugation, and how the components were separated. This is the Hershey and Chase's experiment. They concluded in 1952 that the genes of these phages are made of DNA, which is a genetic material, and is transferred from virus to bacteria and not protein. So DNA and not protein is the genetic material. It was later Avery, McLeod, and McCarty's experiment in 1944 of isolation and establishing the transforming principle that Frederick Griffith identified. To determine the biochemical nature of this transforming principle, they purified DNA, RNA, and proteins from the heat-killed S3 strain, that is the fourth set of strain, and determined which of the above molecules converted the R strain into the S strain. So for their experimental setup, they took the fourth set of Frederick Griffith, that is a heat-killed S form, extracted it in a liquid medium, centrifuge, heating, homogenizing, and getting a cell-free extract. Now, they divided this into 4 sets. Set 1 was the same set that was used by Frederick Griffith. He transferred into R-type culture, injected into the mouse. The mouse died. In set 2, they treated with RNAs to remove the RNA, transferred into R-type culture, injected into the group of mice. The mouse died. Then the set 3, they treated it with protease to remove the proteins. The mouse transferred to R-type culture, injected into the group of mice. Mouse dies. In the last set, they treated it with DNAs to remove the DNA and then transferred into R-type culture and injected into the group of mice, and the mouse lived. Now, they observed that the transforming principle in S cell was not destroyed by heat, not by RNAs, not by protease, but its transforming activity was destroyed by DNAs, abolishing transformation. So they proved that transforming principle was not protein or RNA, but it is indeed the DNA that transformed the non-virulent bacteria into the virulent form. Students, the above 3 experiments, Frederick Griffith, Avery et al., and the third experiment, T2 on the bacteriophages, Martha and Chase, were concerning DNA as a genetic material, wherein Frankel Conrad and Singer's experiment in the year 1956 showed that RNA is the genetic material. In this, they use tobacco mosaic virus, which infects tobacco plants. And this tobacco mosaic virus has a single long RNA molecule, and it's covered with a protein coat. Now, prior to Frankel Conrad doing the experiment, it was Frankel Conrads and R. Williams who showed that a virus can be separated into in vitro into its component parts and can reconstitute a viable virus. So this finding led Frankel and Conrad Singer, who constituted TMV with parts from different strains. So they performed their experiment. They took two types of RNA viruses, that is TMV and HRV, that is Holmes Ribs grass virus Plantago strain, and created a new hybrid virus, taking a protein coat from HRV strain and RNA from a common TMV strain. Different other strains were also used and produced different recombinant strains. Then these strains were allowed to infect by rubbing this hybrid virus separately on the leaves of tobacco plant. This chart to show you how they did the TMV reconstitution experiments. That is, they took the TMV, the RNA from the TMV and the protein coat from the HRV strain and reconstituted. Once they rubbed it against the tobacco leaves separately, they saw a TMV progeny. Now, TMV produced during the process of infection that developed exhibited the symptoms of the disease typical of the strain from which RNA was taken and not the protein. Hence, the infection produced was of the type associated with the RNA and not with the protein. This proved that specificity of proteins was determined by RNA alone and not proteins carried no genetic information. Nucleic acid, that was RNA, was shown to be the genetic material. These are the references, students. These are the online references. And thank you, students, for a patient hearing.